Hello and welcome to the Biological Anthropology lecture series. I'm Dr. Richard Jonathan O. Tadurin. Here we explore what it means to be human, biologically, evolutionarily, and behaviorally. Let's begin our journey into the science of humanity. Welcome to Module 4, Primatology. Here we study our closest living relatives, their anatomy, behavior, and evolutionary significance. In this section, we examine primate taxonomy, from strepsirrhines and haplorrhines to Old World monkeys, New World monkeys, and apes. Let's revisit where primate diversity begins. The major split between strepsirrhines and haplorrhines. This division isn't arbitrary. It reflects deep evolutionary divergences that shaped how different primate groups sense their world, move through their habitats, and interact socially. When we talk about these two suborders, we're really talking about two different pathways primates took in adapting to their environments. Let's look at strepsirrhines first. These include lemurs, lorises, and galagos, and they retain several ancestral features that give us a glimpse into early primate evolution. One of the most striking is the rhinarium, the moist, hairless nose associated with a strong reliance on smell. You might wonder why scent matters so much. In many nocturnal or crepuscular environments, smell becomes a powerful way to navigate, communicate, and locate food. Strepsirrhines also have traits like a grooming claw and a tooth comb, which serve both social and functional purposes. Their eye orbits are less completely enclosed, and their brain-to-body ratio tends to be smaller. Again, reflecting sensory priorities shaped by specific ecological pressures. Think about this. If you're active at night and moving through dense vegetation, your sensory toolkit will evolve very differently from that of a diurnal species swinging confidently under full daylight. The shift into the haplorrhines marks a different evolutionary strategy. Haplorrhines include tarsiers and the anthropoids, and they are characterized by a greater emphasis on vision. Their eyes are usually fully enclosed by bone, offering better protection and stabilizing the visual system during movement. They lack the rhinarium, signaling reduced reliance on smell and increased reliance on sight. Tarsiers are especially interesting here. They are nocturnal like many strepsirrhines, but their visual adaptations, enormous eyes for one, show how different evolutionary routes can arrive at similar solutions. It's a reminder that classification reflects ancestry, not just appearance. As we move from tarsiers into the anthropoids, monkeys, apes, and humans, we see the continuation of this pattern. Larger brains, more complex social behavior, and sensory systems aligned with diurnal lifestyles. But for now, focusing on the strepsirrhine-haplorrhine divide reminds us that evolution often works by modifying existing structures rather than inventing entirely new ones. You might find it helpful to think of these suborders as two branches on a tree, each experimenting with different combinations of traits. So as you study primate diversity, keep asking how these differences arose. What environments favored strong olfaction? What challenges pushed other groups toward enhanced vision? These questions help us move beyond memorizing traits and toward understanding the evolutionary logic behind them. By the time we reach the anthropoids, this foundation will make the next layers of classification feel much more intuitive. Let's revisit where anthropoids branch out in the primate family tree. Once we move beyond the tarsiers and into the monkeys and apes, we see an explosion of diversity shaped by geography, climate, and the daily challenges of life in different environments. Today, we're focusing on two major monkey groups, the New World monkeys of the Americas and the Old World monkeys of Africa and Asia. At first glance, they all look monkey-like, but once you slow down and look carefully, their differences tell a much deeper evolutionary story. Let's look at the New World monkeys, or platyrrhines. These primates evolved in the forests of Central and South America, settings where a largely arboreal lifestyle shaped their anatomy and behavior. One of the most noticeable features is their outward-facing broad nasal openings. But what often captures students' attention is the prehensile tail in several families, especially the attalids. You might wonder why a tail would become so functionally important. In dense rainforest canopies, a strong, grasping tail becomes almost like a fifth limb, helping stabilize the body during feeding or fast movement. Their dental patterns and diet show a similar story of adaptation to rich, varied forest resources. When we shift to Old World monkeys, or catarrhines, we're encountering primates shaped by very different ecological pressures. These monkeys inhabit not only forests, but also savannas, highland environments, and even human-dominated landscapes. Their downward-facing nostrils are just one anatomical cue separating them from their New World relatives. More revealing is their dentition. Old World monkeys have bilophodont molars with ridges perfectly suited for grinding tough plant material. Think about this the next time you see a macaque or baboon calmly chewing. It's a reminder that teeth often map directly onto diet. You might also notice that Old World monkeys show considerable sexual dimorphism. In many species, males are larger, with more robust canines, reflecting social systems where competition plays a prominent role. This contrasts with many New World species, where dimorphism tends to be less extreme. Locomotion also differs. Catarrhines generally lack prehensile tails and rely instead on a variety of quadrupedal movements, sometimes supplemented by terrestrial foraging. Even though both groups are monkeys, they represent separate evolutionary experiments. Their similarities reflect shared ancestry within the Anthropoids, but their differences reflect millions of years of living in distinct ecological worlds. You might wonder why this comparison matters for understanding human evolution. The answer lies in recognizing patterns, how geography shapes anatomy, how diet influences teeth, how social systems influence body size. These are the tools we use when interpreting fossils and positioning early hominins within the broader primate story. So as we move forward, keep asking what each anatomical feature or behavioral trait reveals about a species' past. In the contrast between New World and Old World monkeys, we see evolution writing two parallel but distinct narratives, each helping us sharpen our understanding of primate diversity and the evolutionary pathways that eventually led to us. Let's revisit where hominoids sit within the primate family tree. Once we step beyond the Old World monkeys, we enter the realm of apes and humans, species that share a more recent common ancestry and a distinctive set of anatomical traits. You might wonder what actually defines an ape. Now this part is important. Hominoids are characterized by features such as the absence of a tail, a broad and flexible torso, and a shoulder built for a wide range of motion. These changes mark a shift toward more versatile locomotion, from brachiation in gibbons to the knuckle walking of gorillas and chimpanzees. Let's look at the teeth, because dentition tells a parallel story. Hominoids have what we call a Y5 molar pattern, where the cusps form a Y-shaped groove. It may seem like a small detail, but this dental configuration reflects shared ancestry and helps distinguish apes from Old World monkeys. When paleoanthropologists uncover isolated teeth, which happens more often than you might think, this pattern becomes a crucial clue for classification. As we consider the different groups within the hominoids, think about the environments they inhabit and the strategies they use to navigate them. The hylobatids, or lesser apes, move with remarkable speed and elegance using brachiation. Orangutans live largely solitary lives in Southeast Asian forests, their bodies adapted for careful, deliberate climbing. Gorillas and chimpanzees, on the other hand, form complex social communities that tell us a great deal about the roots of human sociality. You might wonder why studying ape behavior matters for understanding humans. The answer lies in the shared cognitive and social tendencies that appear across these species. Cooperation, tool use, hierarchy, conflict resolution, these behaviors remind us that the foundations of human social life are not recent inventions, but deeply embedded evolutionary patterns. Now, this part ties everything together. Humans are hominoids too. While our lineage took a dramatic turn with bipedalism and later with cultural and technological innovation, we still carry the anatomical and behavioral echoes of our ape relatives, our flexible shoulders, our dental patterns, aspects of our social behavior. All of these reflect a shared evolutionary heritage. So as you think about hominoids, don't see them as a separate category of exotic animals. See them as living branches of the same evolutionary tree that produced us. Their anatomy and behavior help illuminate what is unique about humans and what connects us to a much older primate story. As we close this section, let's revisit the big idea that ties all these groups together. When we move from strepsirrhines to haplorrhines, from New World monkeys to Old World monkeys, and finally to the apes, we're not just listing categories, we're tracing evolutionary pathways. Each group reflects a different set of solutions to life's challenges: how to find food, how to move through forests, how to communicate, how to raise young. You might wonder why these distinctions matter when we're ultimately interested in human evolution. Now, this part is important. Understanding the diversity within primates gives us a framework for understanding where humans fit within this broader family. Let's look again at how geography shaped these evolutionary branches. Primates in the Americas had very different ecological pressures compared to those in Africa and Asia, and their anatomy tells that story clearly. Meanwhile, the transition from monkeys to apes highlights shifts in locomotion, diet, cognition, and social complexity. These aren't arbitrary differences. They're evolutionary experiments that help us interpret the fossil record and make sense of early hominin traits. Think about this the next time you see a primate in a documentary or a nature reserve. Its nose shape, teeth, tail, or lack of a tail are not quirky features. They're clues to a lineage's story, and by learning to read those clues, we become better equipped to understand the origins of our own species. In the next section, we'll shift from classification to behavior, exploring how primates live, cooperate, compete, and communicate in their daily lives. Thank you for joining this lecture. I'm Dr. Richard Jonathan O. Tadurin. I hope this session deepened your understanding of biological anthropology. Keep exploring what it means to be human. See you soon.